Oh, we recording? Sweet. Alrighty. So, welcome back to another fun biochemistry 2 video. Today we're actually going to be covering a lot of content. I didn't realize how fast Dr. G was moving along. So, we are going to be starting around end of page 16, and we are going to be going all the way down to about page 24. So, we're covering I don't even know. Basically, malate shuttle. We'll talk about a little bit about carbo digestion in the liver. We'll be talking about the malate shuttle, creatine phosphate system, Cori cycle, lipid digestion, lipid absorption, beta oxidation, and ketone bodies. So we have a lot to cover today. So this will probably be a longer video. I'm just going to try and fly through it and give you guys the basic overview again. But let's get into it. So starting off here, we're talking about carbohydrate digestion in the liver. And if we remember, the liver has some cool functions when it involves glucose. We can go through gluconeogenesis, and we can also go down the pathway of breaking down glucose as well. But some cool things that we need to know, particularly about the liver, is that when we are going from glucose to glucose 6P, instead of it being hexokinase, similar to muscle cells, we are going to be referring to this enzyme as glucokinase. And again, costs us 1 ATP right here to transform glucose into glucose 6P, but just a different enzyme there. And if we remember too, we can also go from glucose 6P back to glucose through the work of glucose 6-phosphatase. And this just allows us to basically remake glucose and allow us to put glucose back in the blood in cases of low blood sugar and situations like that where our body signals the need for glucose to be put back into the blood instead of just being stored in the liver. And that mechanism right there of being able to take glucose and put it back into the blood is very specific to the liver, sometimes in extreme cases kidneys, or actually kidneys have a different function. But specifically for the liver, just understand that glucose can go back and forth between the blood plasma pretty easily. And then we are going to be talking about enzyme efficiency. So what we need to talk about is glucokinase. This enzyme right here has a very high Km, which signifies that it has a very low affinity to initiate a reaction. So if we only have like one, I don't even know that metric. I'm just going to call it millimoles. If we only have one, this enzyme is not going to be very inclined to function to convert glucose into G6B. We need a very high concentration of glucose to begin with to allow this enzyme to activate. So we need to have a huge abundance of glucose to initiate, like, hey, we want to start transforming these glucoses into glucose 6P and so on and so forth. So it is going to be kind of difficult to get this enzyme to react unless we have a super high concentration of glucose already prevalent. And then if we look right here, glucose 6-phosphatase, or our enzyme that converts glucose 6P back into glucose, has a little bit of a smaller Km, so means it's going to have a lot higher of an affinity to activate and allow glucose 6P to transform back into glucose. And this is kind of like a roadblock mechanism here. So let's say we have a ton of glucose and we activate glucokinase and we get that glucose to convert into G6P, but our body doesn't need to break down glucose 6P, and we just start getting a high amount of glucose 6P just sitting there. Well, this enzyme, glucose 6-phosphatase, is immediately going to sense that. And as soon as we hit this amount right here of glucose 6P for this enzyme to activate, glucose 6-phosphatase is immediately going to say, hey, I have a high concentration of glucose 6P that I really don't want sitting there. I'm going to revert it back to glucose, and then it will go from there, either back into the blood, or it'll just sit there and maybe just wait until we need to utilize glucose a lot more. Yeah, and we just have a little bit of a list right here. Usually in the questions, you guys are going to see these of KMs and affinities are going to be listed, and basically what Dr. G is looking for is you guys are able to look at find the right enzyme that's going to work for what it needs. So if it says glucose, there's a high abundance of glucose 6P. He kind of wants you guys to understand that glucose 6-phosphatase is probably going to act on that, and we need our glucose 6 beta concentration to be equal to this Km or around that Km for this mechanism to even begin. Again, go look at the questions. You guys are going to be building good bridges of knowledge rather than just having me lecture to you here. But let's look at some other stuff. So glucose 6 kinase is similar to an allosteric enzyme, and we are inhibited by high levels of glucose 6p. So again, if we have a huge roadblock of glucose 6p here and we are not utilizing it, glucokinase is not going to want to break down any more glucose, because then we're just going to be adding to that surplus already. We're going to wait until that glucose 6p is either reverted back to glucose or further broken down in the pathway. And once these levels of glucose 6p get polished out, glucokinase is going to activate again and allow for the breakdown of glucose. And then I know I mentioned gluconeogenesis very briefly, regulated by the hormone glucagon. This is exam 3 knowledge, but it's basically just a hormone that helps regulate blood sugar. Yep, talked about the low Km of glucose 6 phosphatase there, and then glycerol phosphate shuttle. We covered this a video or two back. So again, just look through it. Understand that we are producing 1.5 ATP per turn, but if you guys remember, everything in the second stage happens twice. So in reality, we are making 3 ATP total. And it only occurs in skeletal muscle under aerobic conditions. So if we remember anaerobic, we have the reduction of pyruvate here that allows us to replenish our NAD plus supply, and that NAD plus will come back. But in aerobic conditions, we do not get that reduction of pyruvate there, so we need that NADH to come in here and react with DHAP to go through the glycerol phosphate system to replenish that NAD plus supply at the beginning of stage 2. All right, let's talk about Malate shuttle. So Malate shuttle is pretty neat, and you can kind of wrap it in more on the edge of the TCA cycle. So this is just another way to oxidize NADH in aerobic glycolysis. And instead of using this shuttle right here, this is just a second mechanism. We do need to know that it only happens in hepatocytes and cardiac cells, and it happens within the inner mitochondrial membrane, basically crossing the membrane here. As you guys can see, we have this little membrane separating the matrix from the mitochondria, or blah, blah, blah. Sorry, I just got all confused there. Basically separating the mitochondria from the cytosol, mitochondrial matrix from the cytosol. So this process is reversible, unlike the glycerol P shuttle, and the name of the game is transforming a lot of these different chemicals or molecules present into different forms so we can cross the inner mitochondrial membrane. So to begin, we're just going to look at oxaloacetate here. Oxaloacetate, we can send that out, and if we remember, it's at the top of our TCA cycle. We need oxaloacetate to combine with acetyl-CoA to get our TCA cycle kicking. But what happens if we have an overaccumulation of oxaloacetate and we don't have a lot of acetyl-CoA coming down? That oxaloacetate is just going to start collecting at the top, and this is just another way to get some NADH, get some energy going, and replenish some NAD plus supplies. So we'll look at oxaloacetate here, and we can see that it's activated or acting with glutamate oxaloacetate transaminase, and this enzyme is basically allowing oxaloacetate to change forms here so it can cross across the inner mitochondrial membrane. So oxaloacetate, when it goes through a transamination reaction, basically we're taking and we're transferring amino groups on some of these molecules here. And what we really need to know, let me erase some of my writing here, is when we were talking about transamination reactions, for our own knowledge, we just need to remember a couple phase changes. So let's refer that to oxaloacetate, and this arrow will just represent transaminase reaction. So oxaloacetate is synonymous with aspartate. When it goes through a transamination reaction, it will transform into aspartate, and likewise for aspartate. If it goes through a transamination reaction, it will go back into oxaloacetate. And then we can also look at alpha ketoglutarate, and that is synonymous with glutamate. And I just like to remember that we just see G and G. That's how I remembered it, and it got me through biochem too. So maybe it'll help you guys with the same trick. Then we also have pyruvate, and this is more seen down in the Cori cycle, which is right here. And we can see pyruvate goes through a transamination reaction and it transforms into alanine. So pyruvate can be associated with alanine. Where was this? Pyruvate right here. Alanine. So anytime you guys see any of these molecules referencing a transamination reaction, just understand what forms they are associated with. Pyruvate goes to alanine, alpha-ketoglutarate goes to glutamate, and oxaloacetate goes to aspartate. So with that background knowledge out of the way, oxaloacetate goes through a transamination reaction, turns into aspartate here, and aspartate is able to cross the inner mitochondrial membrane, so we can track it across. It will go through the inner mitochondrial membrane, enter the cytosol, and aspartate here will go through another transamination reaction. And if we follow the arrows, aspartate will turn back into oxaloacetate here. And we can see that with this transamination reaction, we also have alpha-ketoglutarate right here. So when alpha-ketoglutarate comes through, it will also turn back into glutamate. And these little arrows right here basically just representing transfers of the amino groups or amine groups. And this is what allows the transamination reactions to happen and these phase changes to happen. So aspartate will come down here and transform back into oxaloacetate. And this is how we get oxaloacetate from the mitochondrial matrix into the cytosol here into oxaloacetate. So now that we have oxaloacetate out in the cytosol, we want to pick up a hydrogen so we can bring it back in the mitochondrial matrix and allow that hydrogen to go through a redox reaction, which we can see over here in this later stage. But how does oxaloacetate pick up that hydrogen? So oxaloacetate is going to come right through here and be acted upon by the enzyme oxaloacetate hydrogenase, and that is basically signifying that oxaloacetate is picking up a hydrogen within the cytosol. So right here, NADH comes out, gets oxidized, and oxaloacetate gets reduced. And when oxaloacetate gets reduced, it just goes back into malate. And if we remember from the TCA cycle, if we do a quick flashback, we can see that this process kind of happens already with malate and oxaloacetate. When malate goes to oxaloacetate here, it loses the hydrogen, and we go through a little redox reaction right here. So when oxaloacetate gains a hydrogen, it's just going to transform back into malate. So that is just how that little mechanism occurs right there. And if we flash back this little picture, so now we have malate, and we know malate is carrying a hydrogen right here. And malate can also go across the inner mitochondrial membrane. So malate is going to come shooting across this membrane again back into the matrix. And what do you know? We're going to go through basically that end stage of the TCA cycle all over again. Malate is going to react with NAD plus here. We are going to reduce NAD plus by adding a hydrogen onto it. Malate is going to get oxidized and turn back into oxaloacetate when we go all the way up here, and we can see the enzyme malate dehydrogenase. It's basically saying, hey, we're removing a hydrogen from malate. And we can look right here, and we produce ATP. We get a little redox reaction down here, and with NADH or niacin, we know we are producing 2.5 ATP per cycle, but since we were in the second stage, in all reality, that is 5 ATP because everything in the second stage happens twice. So the malate shuttle is just basically allowing oxaloacetate to change forms, exit the mitochondrial matrix, enter the cytosol, pick up a hydrogen out in the cytosol, and then transform back into malate, go back across into the mitochondrial membrane down here with that hydrogen, and it allows us to produce a little bit of ATP here and get a change of hydrogens going across the inner mitochondrial membrane to allow the fuel for that little redox reaction to happen right there. Let's see what other big stuff, again, hitting on hepatocyte and cardiac cells. Yep. Yeah, so basically this little tidbit of information right here is just comparing the malate shuttle and the glycerol P shuttle. We know that the glycerol P shuttle involves riboflavin or FADH, and FADHs, when they go through redox reactions, we know we're only producing 1.5 ATP or 3 per 2 cycles. And the malate shuttle uses niacin instead, and with the NADHs and everything, and we produce 2.5 per cycle, and in all reality, per 2 cycles, we produce 5 ATP. So this is how you could get a little bit of a different ATP count right there. So now let's talk about the creatine phosphate system. And this is just another way for our body to store energy, and this kind of helps out or goes along with glycolysis to an extent. If glycolysis is limited by allosteric inhibition and our enzyme back from the first stage, PFK, is shut down, this is just another way for our body to help get some energy. So Let's look at this. Okay, so this is the main little diagram that we want to know right here. And if we look, we have a couple ATPs being used, ATP being produced, and we also have a phosphate group leaving there. So the main enzyme that or molecule that we're going to be talking about is creatine. And we need to get creatine into a storage form where it's holding a phosphate and allows us the potential to make energy. So creatine is going to react with an ATP here, and it's going to cost us one ATP to get creatine into the storage form of creatine P. So use one ATP, our count is -1, and we're sitting here at creatine P. So we have two situations on basically determining which way we can go with this reaction here. The first way we can go is if you're being active. So if you have some creatine in your body, you add on a phosphate group to it, make some creatine P, and then you just go for a run, do some awesome weightlifting or something like that. Our body is going to go through this lower process right here. And basically what's going to happen is creatine P is going to realize, hey, we need to make some energy. He's being active. We need to help out, give some ATP. So we're going to have an ADP come up here and bind with this. We're going to have creatine P react with ADP here, and we're going to produce an ATP. So right here, we are +1. And then, as you guys can see, we get creatine again, and this creatine is going to go back and be recycled. And so it's just going to come back up here and go through this whole process again of getting to the storage form of creatine P. Now, the other pathway is basically signifying if you're not being active. If you, I don't know, someone brought up a good question about creatine monophosphate or supplements you can take. I don't know enough about biochemistry to speak on that, but for this specific instance, we're just talking about if you have creatine P storages and you're not active and you just sit on the couch and don't do anything, you are actually costing yourself energy. So if you have a huge storage of creatine P or you're building up that storage and you're not active and you just sit on your butt, you're going to go through this upper pathway up here and you are going to transform creatine P into creatinine. And then that creatinine is going to go and be excreted from your urine. But if we look, H2O is coming down here and helping remove the phosphate, but we're not producing any ATP. So if you are adding to your creatine P storages and you are not being active, you are costing yourself energy. You are not producing any energy. And this creatine is just going to be excreted out through your urine. So you are actually decreasing your creatine storages to go through this whole beginning process right here. So if you're building up your creatine P storages and you're being active, your ATP counts are kind of balancing out and you're recreating creatine to come back up here and restart the cycle. But if you have creatine P storages and you're not active, you're not producing any ATP. In the second step, you're transforming into creatine or creatinine, and then you're just excreting it through the urine. So that is a little bit more of an unhealthy and less efficient step. And another little side note or historical note, most of the times that the questions referring to this mechanism come up, Dr. G will be more or less referring to this little mechanism right here. So if you see creatine P and an H2O involved at all, you should immediately be thinking your products are going to be a phosphate group and creatinine right here. Now, if it doesn't mention H2O and it mentions someone's being active and everything, that's when you think about this bottom stage right here. But unless he specifies it, more or less have this upper mechanism in your mind. And then this little equation right here basically is just doubling down on what I said about exercising with creatine P. As exercise increases, our creatine P levels right here are going to decrease, which makes sense because we're using up that supply of creatine P. And our concentration of creatine is going to increase, which again makes sense. When we're active, we're using up the supply right here. We are producing ATP and we are creating a higher concentration of creatine in the second stage. I'd say just these amino acids right here, the glycine, arginine, and methionine. These are essential amino acids associated with creatine. So I would just have those in the back of your mind. Again, I haven't seen too many questions referring to amino acids being involved with this. However, you never know what type of question you're going to get. So after you get a lot of these overarching concepts down, come back and look at a lot of these minor details. again, this is just a good side note about creatine being excreted. So again, if you're not active, you'll get creatine. It'll be excreted through your urine compared to if you are using creatine P for activities, you're going to get this equilibrium going between creatine and creatine P because you are replenishing your creatine storages. Um, all right. Honestly, I think that's enough. So let's start talking about the Cori cycle. The Cori cycle is really interesting, and it's a process that allows us to take lactate from anaerobic glycolysis or respiration and convert it back into glucose to re-enter glycolysis as glucose 6P. And when you see the words recreate or convert back into glucose, your first thought should be the liver, because the liver is one of the few organs in the body that can actually do this whole process. This usually occurs after exercise, during rest, because after rest, I mean, we are running through glycolysis so much using these carbs for energy, trying to produce energy, but when the system shuts down, we might get an overabundance of lactate here. So this is a system that allows us to use this surplus of lactate to help function and supply some other processes inside our body. So basically what's going to happen here is we're going to go through anaerobic respiration or glycolysis or whatever process we're talking about. We're going to come down here, glucose 6 phosphate, we're going to turn into pyruvate, and then what we know with anaerobic respiration is we're just going to go from pyruvate into lactate. And with lactate here being present in skeletal muscle, we are going to be able to transfer it into our liver through our blood. So we are going to take lactate, transfer it through our blood, and pop it into the liver. And as soon as we hit the liver, we can go through gluconeogenesis or creating glucose or G6P from later stage steps. So lactate is going to transform back into pyruvate. Pyruvate is going to come back and transform back into glucose 6-phosphatate. And I always like glucose 6-phosphatate because it has so many different functions on the pathways that we talk about. We can convert it into glycogen. We can honestly just turn it back into pyruvate if needed, or we can even revert it all the way back to glucose. And if we remember, the enzyme that allows that to happen is G6-phosphatase, or basically just removing that phosphate group from glucose 6P so it can be turned back into glucose. And then little side notes here, this is gluconeogenesis, the recreation of glucose. And then how we talked about transamination reactions earlier, we also have pyruvate here who can be acted on by a transamination reaction, turn into alanine, and alanine can come all the way into the liver, go through another transamination reaction to turn back into pyruvate, and pyruvate can re-enter the cycle in the liver as well. So we have a couple different mechanisms going on here. And then there's also an example question about the efficiency of this system, because this system is not 100% efficient. We are losing some sources of glucose or some sources of carbon to carbon dioxide or CO2. And basically, this example question is basically saying is if we have 150 moles of glucose that produce 2 lactate each, which we know that as soon as everything hits the second stage, it happens twice. So per 1 glucose, we're going to produce 2 lactate. And if we have this extra abundance of lactate, theoretically speaking, when this lactate comes back through and goes through the Cori cycle all over again, theoretically, we should get the same amount of glucose back for the lactate we have, but in all reality, it doesn't happen that way. And about 20% of our glucose that's used is going to actually be turned into carbon dioxide. So all we're going to do here is basically times 150 glucose by 0.2 or 20%, which should give us about 30 millimoles of glucose. And we just subtract that from the overall total. So when we go through the Cori cycle, in all reality, if we put in 150 moles of glucose, we are only going to get about 120 moles back through the Cori cycle. Just looking for any little small notes here, kind of doing this on the fly. No. All right, so now I'm done with the Cori cycle here, and we are going to move into lipid digestion, and then following that, we have triglycerol activation, some beta oxidation stuff, which I have a little bit of a easier way to go about that. And then we'll just talk about ketone bodies, which ketone bodies are pretty simple, and I personally really like this diagram, so we'll be talking more about that diagram right there. And someone mentioned to me you guys made it all the way to ketone bodies for quiz content, but no one was even sure on whether or not you guys are going to be quizzed on it. So I'd rather cover it now just in case, because you never know what type of questions you're going to get. So we'll talk about lipid digestion here. And basically what I'm going to do is I'm going to walk through all the text here first. I'll draw out a little diagram just to get a better visual for you guys so we can better understand what's going on. And then we'll just keep on moving. So let's start at the beginning. In the mouth, we eat lipids. We ingest lipids, and in the mouth, no true digestion is happening. So don't get tripped up with that. We are not truly digesting anything in the mouth. However, we are secreting enzymes to attach to our food so they can be activated later on in the system. So lipids are hydrophobic, so the saliva in the mouth doesn't interact with the fats. And the biggest thing that we are getting secreted in the mouth are lingual lipases. And these enzymes are just being secreted from the mouth to attach to our food, and they will eventually be activated later in the stomach, but we're just getting all the enzymes on the food in the mouth. And we also go through this thing called mastification, which is basically just chewing up the food. And what this does is this increases the surface area of the food in our mouth. And it allows us basically to create a greater surface area of this food to allow more enzymes to attach and allow greater enzyme efficiency later on once they become activated. So once we, and it does mention here that lingual lipases hydrolyze triglycerols into diglycerols and free fatty acids, and it goes into a little bit more information here. I would recommend just reading that and getting more, once we get this concept down, coming back again and reading through it. But lingual lipases, again, are not activated until they hit the stomach. They are activated by the low pH in the stomach, and that's where the first true digestion happens of the lipids. So we'll hit the stomach, we'll get some digestion. I believe HCl is the main acid present in the stomach that allows for the activation of lingual lipases, but lingual lipases do not activate until the acidic environment. I know I'm repeating myself a lot, but it is just a big concept to know. So once lingual lipases are activated, they start partial digestion in the stomach. The chyme, whatever we call it, with this little mush of food that we have going, it is eventually going to pass from our stomach into our small intestine or into the intestinal lumen. And this is where we get a second enzyme present. We get pancreatic lipase. And pancreatic lipases breaks down triglycerols by hydrolysis, and it basically allows for increased surface area, and we get even more products. We get free fatty acids, we get two monoglycerols, and we get some cholesterol present too. I always just like to say that pancreatic lipases are the main digesters of food because they just act a little bit more efficiently, because lingual lipases are definitely acting and helping with digestion and getting a good amount done, but pancreatic lipases really come in and pick up a lot of the scraps when digesting lipids. And this little information is a little bit all over the place. So we'll just talk about these two hormones that are secreted and how they interact with some of the enzymes present. So we'll talk about cholecystokinin. And basically cholecystokinin, it's telling our body that we should release bile. And it sends a hormone. Hormone somehow gets in contact with the gallbladder, and the gallbladder releases the bile. And if we remember, the liver creates bile, the gallbladder just stores it and allows it to be released into the intestinal lumen. Um, and the main function of bile, what it really helps with, is it slows the motility of the chyme or whatever we want to call it. And so when this chyme enters the intestine, the release of bile will help slow it down and allow a longer period of time for our enzymes to act upon it and help break it down even further and just help the enzymes work at an efficient rate rather than having this just flow through our small intestines and only being able to digest like 50% of it. What we should know is bile. Ooh. Sorry, I just had a brain fart there. We should just know that secreted by the cells of the duodenum and upper jejunum. Um, yeah, we just need to know locations and everything like that. So cholecystokinin, I believe that's where it's secreted. I can't remember if it's talking about bile there. I apologize. I'm going to go look that up later and get back to you guys on that. I really should do some research ahead of time to know some of these answers. But besides cholecystokinin, for cholecystokinin, we just need to know it helps release bile and it slows down chyme to allow these enzymes to act more efficiently. So now we'll talk about secretin right here. And this enzyme causes the liver and pancreas to release bicarbonate solution And what's really cool is pancreatic lipases do not work well in an acidic environment. They are not overly efficient in acidic environments. So when this chyme passes from our stomach into our intestine, we still have some acid present, and it's keeping the pH of that area still pretty low. So pancreatic lipases, when they're released, they can act and slowly start digestion, but they won't truly start running super efficiently until the pH is raised. And that's where secretin comes into play because it helps release bicarbonate into the intestinal lumen and allows these pancreatic lipases to really start kicking off. And they're released by intestinal cells in response to low pH of the gastric fluids. Um, and it just helps promote the release of bicarbonate. So with this little tidbit right here, I believe cholecystokinin is released by the cells of the duodenum and upper jejunum. And that's how these two enzymes work together. Cholecystokinin is more for bile release. Secretin really helps with bicarbonate release to raise the pH in the small intestine and allow our pancreatic enzymes to work more efficiently. So if we were to do a quick drawing real quick, I did this drawing the other day in tutoring, but basically we'll start up here, have the throat, stomach, and I'll just somehow draw a mouth right here. I'm not an artist, I can tell you guys that much. So let's have our mouth right here. We'll ingest some lipids. We'll get the release of lingual lipases in the mouth. We also have mastification going on here, which is increasing the surface area of our food and help breaking it down, allowing lingual lipases to bind to more areas of that food. And then that chyme is basically going to come down here into our stomach, and due to the low pH of our stomach, our lingual lipases are going to become activated. And partial digestion is going to start happening in the stomach. And then we'll get down here to the small intestine. And that's a little valve right there. And once the food moves from the stomach into the small intestine, we'll have the release of cholecystokinin. I'm just going to put CK right there, which is going to be helping with bile release. And what all that is doing is slowing down the chyme. And then we're also going to get the release of secretin, which is raising the pH, and it's helping activate pancreatic lipases. So in here, we'll have pancreatic lipases really acting on. Oh man, I'm horrible at spelling today. Pancreatic lipases activating on there, further breaking down food. And again, they are highly efficient at increased pH, so that's why bicarbonate is released. And we will be going down, oops, sorry. We are actually, that's how we break down food in the small intestine. I apologize. I'm a little bit tired today, not as energetic, so I think I just need to drink more water, drink some more creatine to get some of these functions going. I'm just pooped today for some reason. But yeah, that's just basically the whole overview of this system right here. And again, please go look at questions. You guys will see how it's referred to in this. You'll see how a lot of these functions go in relation to the malate cycle, Cori cycle, and even lipid digestion here. But I'm just hitting the need to know topics on the functions that we need to know. And a little bit of the smaller information is more in the hands of just reading through it, reading through the slides, reading through all these different packets, and just developing a better understanding. So yeah, for this instance, this is really all we need to know. So yeah, just a quick overview of the products again of pancreatic lipases. We have free fatty acids, 2 monoglycerols, and cholesterols. And then up here, we do get our products with lingual lipases as well. And then now we're going to start moving into lipid absorption. So once digested, the products of the pancreatic lipases have two different fates. So we have two different fates between long chain fatty acids and short and medium chain fatty acids. So we have 2 monoglycerol as well included in this long chain fatty acid category, but the long chain fatty acids and 2 monoglycerols are converted to triglycerols, which are then transported through the body in the form of a chylomicron. And the chylomicron right here is basically just a transport to move these triglycerols around the system and to put them to different functions to get activated, which we can see right here, activating the activation of triglycerols, but we'll get to that in a second. So we just really needed to associate that long chain fatty acids and 2-monoglycerols are stored in chylomicrons until they're released somewhere else in the body, and they can also be thrown around through the lymphatic system. And then we'll talk about short and medium chain fatty acids. These basically just go through the hepatic portal vein to the liver and are converted into triglycerols for storage. We just need to differentiate between long chain and short and medium chain, and we will be A-OK. So we are going to be moving on with more long chain fatty acids, and they are transported through chylomicrons. So where do we go? These chylomicrons will eventually go through the lymphatic system, and we will go into the cytosol cells and actually eventually to the mitochondria to initiate the process of beta oxidation. But how do we activate them? And actually, what was really interesting the other day is I was tutoring someone, and this packet I have here, I didn't realize there's different versions of this packet, because the packet that he had, in this ATP count right here, it was only one ATP, and up here it only mentioned only one phosphate being dropped off. However, I learned it as 2 phosphates and 2 ATP are used for this whole process. So I will do some deep diving more on the function. I'm believing that this whole system is referencing it just running 2 times through, which is why we use 2 phosphates for the activation. It's either that or we need 2 acetyl-CoAs or something like that, but I will lock down on some better answers for you guys. But we'll just cover the main mechanism that we need to know, because there are just some questions that get into the specifics of this system, and we will cover that. So once this chylomicron is activated, this long chain fatty acid is activated, we are not activated, transported. We're going to start activating some triglycerols here, and we're going to get them prepped and ready for beta oxidation. So we can see right here that we start off with a fatty acid, and this fatty acid is going to be acted upon by ATP and our CoA enzyme right here, which I believe, where's our little B vitamin chart? That was coenzyme A, which should be pantothenate or vitamin B5. So keep that little tidbit of knowledge locked in your brain there. So we can see right here, fatty acid, whoop, activated by gets 2 phosphates dropped off, gets CoA dropped off, and it's acted upon by the enzyme thiokinase. So once we get acyl-CoA is going to go from the cytosol here and go across the membrane into the intermembranous space of the mitochondria. And we can see we have a little couple functions here. So this acyl-CoA is going to combine with this other little molecule right here called carnitine 1. And what this is basically allowing it to do is that fatty acid really can't, or acyl-CoA really can't pass through this membrane in here, because acyl-CoA cannot cross the intermitochondrial membrane, and we need it to get through here. So we are basically allowing it to change form, similar to the malate cycle or the malate shuttle when we are transaminating oxyl acetate to get it through and across the membrane. The same mechanism is happening here. So acyl-CoA is going to combine here with carnitine 1, and we're also going to lose a CoAsh here in the process. And we are going to have the product called acylcarnitine, or referenced as carnitine 2. And with this different state that acyl-CoA is in, or acylcarnitine, this allows us to get the acyl molecule across the intermitochondrial membrane into the mitochondrial matrix, and is acted upon the enzyme translocase here when acylcarnitine or carnitine 2 is able to cross through this intermitochondrial membrane. And we can see right here, we still have acylcarnitine, and when it goes through this little mechanism right here, we are going to get a couple products. It's going to be acted upon carnitine palmitoyltransferase number 2, or CPT2. And basically what this is doing is we're trying to get this acyl-CoA isolated again. So we are going to add a CoAsh to this acylcarnitine, and that's going to allow us to recreate acyl-CoA over here, because we saw it was here. We need to put through some phase changes, whoop, all the way over here, and this is how we get the acyl-CoA inside the mitochondrial matrix, and we can see that we reproduce a carnitine 1 again, and this carnitine 1 is going to be acted upon by transferase and pop right back into the intermembranous space to get this whole mechanism started all over again. So main products of triglycerol activation is just trying to get this acyl-CoA all the way into the mitochondrial matrix so we can progress into beta oxidation. And we're just talking about the transfers of one acetyl-CoA here, and we use 2 ATP because we were dropping off a couple phosphates over here. And then also, anytime you guys see acyl, we just need to know that a fatty acid is involved somehow. And just a little tidbit of information here, just kind of have a basic understanding that long chain fatty acids are going to be between 12 to 20 carbons on those long hydrocarbon tails. So now we're going to get into beta oxidation. And beta oxidation for me, when I took this quiz, I did not have a good understanding of it at all. And my roommate at the time taught me a really cool trick on just how to navigate this. And all we're going to be doing is looking at the facts. So I'm just going to read through a couple of this information. This stuff over here, if you guys want to learn it, I'm never going to tell you not to learn it because, again, you never know what questions you're going to get. But when I took Biochem 2, I really did not know any of this, and I just looked at the raw data that we had. So little things about beta oxidation that we need to know. It occurs in the mitochondrial matrix, which made sense right here because we were trying to get this acyl-CoA into the mitochondrial matrix. It is strictly aerobic, and we really don't make any actual ATP in beta oxidation, but we supply all the molecules, or we just supply so many products that can eventually be made into ATP, and you guys will see what I mean when I get more into it. But beta oxidation alone really does not produce any. It really doesn't produce any ATP on its own. But what we really need to know is with these long fatty acid chains, every time we go through a cycle of beta oxidation, we are cleaving 2 carbons from this chain to use, and I will go through that as well. And it makes sense too, because the longer the fatty acid chain we have, the more products we can make to eventually be made into ATP. So the shorter fatty acid chain, we really don't have the potential to make as much energy compared to a longer chain. And then after so many cycles, the acetyl-CoA only has 4 carbons left, and the molecule is split into 2 molecules of acetyl-CoA, and beta oxidation is finished. So this little mechanism of 7 cleavages per 8 acetyl-CoA. I will get into that. Actually, one more thing we got to cover. So I will go over this number of molecules of CO2 produced and everything as well. I want to make sure you guys have the full picture ahead of you guys and can think even deeper on some of these concepts. I do not remember really looking at this stuff right here. I'll be completely honest with you guys. So again, if you guys want to go back and look at it, feel free to develop a good understanding of it. It's just I did not learn it at the time, and I don't think I could talk about this as accurately as I can about some of this other stuff. So I don't want to confuse any of you guys. And there are some other phenomenal tutors on campus I'm sure are really comfortable with this concept. So that's a good time to go talk to them. So when we were talking up here, we were talking about more even number carbons or saturated fatty acids with an even number of carbons. So like 16 carbons, 18 carbons. And we know that we cleave in twos. So what happens when we get an odd number of carbons? Well, we are going to be talking about propanol-CoA. So this process is the same basically as even number of carbons, except we're going to be left with 3 remaining carbons instead of just having everything move efficiently and having those just last 4 carbons split into 2 and go off on their separate pathways. When it talks about odd number of carbons, we're going to be using this mechanism of propanol-CoA because we'll have 3 carbons remaining at the very end after going through all these beta oxidation and cleavages. We're going to have 3, so these 3 carbons will eventually turn into propanol-CoA, and they will progress eventually into succinyl-CoA, which, if we remember, succinyl-CoA right here, whoop, that is present in the TCA cycle, and it will also produce 5 ATP eventually in there because we get 1.5 right here, 2.5 right here, and we eventually get a GTP or ATP, another little form of energy right there. Yep, that's a little math right there. All right. B vitamins. I say memorize, lock these in right here. All carboxylation reactions or adding of CO2 require the help of B vitamins. So propanol-CoA changing forms into methylmalonyl-CoA requires biotin or B7. Then L-methylmalonyl-CoA to progress into succinyl-CoA will require the help of cobalamin or vitamin B12. So let's get into the math because I really like the math on this. So basically, if you guys want to look at this from a very overly simplistic way of point of view, you're going to be getting a lot of questions about either odd, or we'll start off with even chain fatty acids or odd chain fatty acids. So even chain fatty acids, we'll just use, we'll use 18 chain fatty acid right here. And if we remember, 18 represents the carbons, and that 0 right there represents the amount of double bonds, and that's more of a biochem 1 concept. But over here on the odd chain, we'll just go with 17 and 0. And if we just want to break this down super efficiently, we're going to try and figure out how much energy or ATP we can produce from these different fatty acid chains. So with even chain fatty acids, our first step before I even get into that, what are we even producing if we break this down? We're going to be producing acetyl CoAs. We're going to be producing NADHs. We're going to be producing FADHs. And we're also going to be able to figure out the number of beta oxidation cycles. So let's break this down. Our first step that we're going to do here for even number fatty acid chains is we're just going to divide it by 2. And this number is going to get us 9, and this 9 is going to start off with our acetyl-CoAs. We are going to be producing 9 acetyl-CoAs per an 18 carbon fatty acid. Now, what's a little bit different is for our NADHs and our FADHs, we're actually going to subtract one more, and per an 18 chain fatty acid, we're going to be producing 8 NADHs, 8 FADHs, and we're going to be going through 8 rounds of beta oxidation cycles. So this 9 right here, and if you guys are confused on why it's 9 instead of 8, if we remember at the end of beta oxidation, we split those last 4 carbons into 2 and 2, and we get an extra acetyl-CoA with it. So that's where this extra little acetyl-CoA comes from. And if we were to break down visually, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. All right, I should have picked a smaller number for that, but per oxidation. So every time we go through a round of beta oxidation, we're just snipping off 2 carbons. So 1 round, 2 rounds, 3 rounds, 4 rounds, 5 rounds, 6 rounds, 7 rounds, 8 rounds. So for an 18 chain fatty acid chain, we are producing, we are going through 8 rounds of beta oxidation to cleave this entire chain down. And with acetyl-CoA, we should also be talking about ATP counts here, because the questions pertaining to this are going to be referencing ATP counts. So we'll put a little thing right here, ATP, and he might even ask you about CO2 counts and oxygen counts here. So I'll show you guys how this all plays in. So acetyl-CoA, we have 9 acetyl-CoAs, and we know that acetyl-CoAs are used for the TCA cycle. And per 1 round of TCA, we know we're producing 10 ATP per cycle. So if we go through 9 rounds of the TCA cycle, since we have 9 acetyl-CoAs, we are producing 90 ATP per acetyl-CoA, or basically in all the sum of acetyl-CoAs that we get from this chain, we are producing 90 ATPs. And if we remember, we produce 2 CO2s per round of TCA cycle, and we consume 2 moles of oxygen per TCA cycle as well. So if we know that we are producing 2 per cycle, we're going to be producing 18 CO2s as well, and we are going to be consuming 18 oxygens as well. Trying to remember if there's any other little tidbit of information we need to know there. No, that should be good. So basically here, this math is, I'm just multiplying 9 by 2 because we know we're producing 2 per. So now let's get into, okay, sorry, I'm all over the place today. Per the TCA cycle and per acetyl-CoAs, this is going to be our main counts and what we need to focus on right here. Per ATP, we just need to know we're getting 10 ATP per acetyl-CoA basically. So if we have 9 acetyl-CoAs, we get 90 ATP. Same kind of mechanism goes for CO2 and oxygen. So now let's talk about NADHs. And when it references NADHs and FADHs, we are going through redox reactions here. So we know per a redox reaction for NADH, we are producing 2.5 moles of ATP per NADH. And for FADH, we are producing 1.5 moles of ATP per FADH. So if I just do quick math, and I wish I was smart enough to do this on my own, we are going to be getting 20, actually I'll do that in better color. We are getting 20 ATPs here per NADH, and we are also getting 12 ATPs here per FADH. Or for all the FADHs we have created, 18 times 1.5 gives us a total of 12 ATP. For 8 NADHs, it gives us a total of 20 ATP. And what we also need to know is we really don't consume any oxygen, or we don't really produce any CO2 with redox reactions. However, we still consume oxygen because if we remember for a redox reaction, when it goes through, we are consuming 1/2 of an O2 per redox reaction to produce CO2. So if we know that, oh, math. If we know we're producing 0.5 per redox reaction, we have 8 opportunities to do a redox reaction with NADH. If we do 8 times 0.5, we know we are producing or consuming 4 moles of oxygen per 8, and we are also be consuming 4 moles of oxygen per FADH. So in reality, in this mechanism right here for an 18 carbon even fatty acid chain, we are going to be looking at about a total of 26 moles of O2 consumed. We are looking at about 18 moles of CO2 produced, and we're going to be looking at about plus 20 plus 12. We're going to be looking at about 122 moles of ATP produced per this fatty acid chain. And I know this is a lot, so again, any questions, please come and talk to me. But I'm just breaking this down because with the questions you get, you will just get raw numbers pertaining to this. And he might ask about CO2, he might ask about O2, he might ask about ATP. So you really just need to read carefully on what he's asking for. But this chart right here and how to do this math, this will be able to help you out for almost any question. So now let's talk about odd chain fatty acids. And we know we have that odd number of carbons of the 3 carbons needed for propanol-CoA at the very end. So how we go about this is instead of worrying about that 3 carbon difference at the end, we just subtract it right off the bat. So we subtract 3 carbons from this because we know it's going to be used. So in all reality, we're looking at about 14 an O chain right here. And if we look at acetyl, NADH, FADH, and number of beta oxidation, our math is going to be very similar. Like everything right here is all going to be the same no matter what, because that does not change. But our numbers of total we have right here does change a little bit. So unlike what we did last time, actually not unlike, we're going to do the same exact thing for the first step. So we're still going to go through the divide by 2, which is going to give us again 7 acetyl-CoA. But instead of subtracting 1 and having 1 less all the way down here, our numbers are going to stay the same all the way through. And if we remember per acetyl-CoA, we get 10 ATP per NADH, 10 ATP or 2.5 per 5 ATP. All of our mechanisms are still going to be the same here. CO2 and O2, and I'll let you guys fill this in if you want. I should probably just go through it just for the sake of it. But a lot of this stuff stays the same from the math aspect. It's just we need to understand the difference between even and odd numbers. We need to know for odd fatty acid chains, we just need to subtract 3 right off the bat to get rid of that propanol-CoA, and then when we divide by 2, our numbers will stay the same, unlike over here to where we divide by 2 and then subtract 1 for all these later reactions. But a lot of our ATP here, the math is still going to be the exact same. Our CO2 is still going to be the exact same. Our oxygen is still going to be the exact same. So we know 3.5, 3.5, da, 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 da. So if we're looking at grand totals here, we're looking at about 98 ATP, we're looking at about 14 of CO2 consumed, and we're looking at about 21 moles of oxygen consumed as well. So this is the very simple breakdown on beta oxidation that I used for biochemistry 2, and it helped me very well, and it got me through the class. So all I'm going to say is you can never really go wrong by using this mechanism here to figure out the math. And there will be some questions that guys do that maybe sometimes the totals don't add up perfectly, and sometimes there's typos in some of the practice problems that we have access to. So just pay attention to that and just make sure your math is right, and make sure to always double check yourself and not just try to do mental math going through some of these problems. Whew, I have been talking for a while here, and I am getting even more and more pooped as I speak. But we have one last thing to talk about, and that is ketone bodies. So, talking about ketone bodies, ketones are produced by the liver in times of carbohydrate shortage. Um, it's basically saying that if we do not have a large source of carbohydrates present, we are going to start producing ketone bodies, and you'll see this a lot on keto diets by reducing your carbohydrate intake. And honestly, if it's taken to the extreme, it is not that great for you. You will go into a state of ketoacidosis, which we'll talk about in a second. But I personally love carbs. I am not going to give up carbs. Dude, I love pastries, man. Like good old donut, or like what are those oh, they're like not cheese. I can't even think of them. They have like, oh man, I'm gonna go to Walmart just to figure out the name, and I'm gonna pick up a pack, and I'm gonna have a sweet treat later. Um but when carbohydrate levels are low, the liver converts oxaloacetate in back to glucose. And if we remember, oxaloacetate is at the top of our TCA cycle, so we're basically taking oxaloacetate and trying to get it back to glucose. And basically what that's going to happen is our acetyl-CoAs that are being used to help be turned into citrates with the oxaloacetates, these acetyl-CoAs are not going to have any oxaloacetate to react with, so our body's going to have to put this towards something else, and it is going to be converted into a ketone so it can be extracted eventually for ATP, and it just gives our body a way to get ATP with these leftover acetyl-CoAs that aren't being used. Um, and the acetyl-CoAs only from beta oxidation can be used in ketogenesis. Lock that fact into your brain. So if you are producing acetyl-CoAs from just carbohydrate ingestion through glycolysis, those can be used for TCA cycle and everything else, but only acetyl-CoAs from beta oxidation can be used in ketogenesis. And we have a couple different processes that we're going to be talking about. We're going to have ketogenesis, which is um, basically the creation of a ketone body, and then we're going to have ketolysis, which is the breakdown of the ketone body for energy. And what we need to know about ketolysis is it occurs in all cells that have mitochondria. So red blood cells will not fall in the category here of ketolysis. And basically, the three ketones we're going to have are acetoacetate, beta hydroxybyruvate (BHB), and acetone. Um, Acetone, in this little fun fact here, it is formed through pathological causes. So for this little picture right here, the main two ketone bodies that we're going to be looking at are acetoacetate and beta hydroxybyruvate, and or referred to as BHB. And well, we can have different ATP counts depending on what we actually go to. So let's get into it. What we do need to know is we need two acetyl-CoAs to even initiate this process. And the act of ketogenesis is only going to occur in the liver. We are only going to create ketone bodies in the liver. So basically what's going to happen is we're going to have two acetyl-CoA come in. We're going to lose a CoAsh here, and we're going to transform into a azoacetyl-CoA. And azoacetyl-CoA is going to be acted upon and be oxidized even more by leaving that CoAsh, and it's going to be acted upon by the enzyme HMG CoA synthase. And it's basically saying we're taking acetoacetyl-CoA and transforming it into HMG-CoA through the enzyme HMG-CoA synthase. And this only occurs in ketogenesis. And this HMG-CoA is going to be broken down into our first ketone body, or acetoacetate. And as we can see, HMG-CoA is almost two products. We have acetoacetate right here, which is a ketone body, and then we can have a leftover acetyl-CoA up here to come back and go through the whole process over again. So on some of the questions you might see about this, don't forget that acetyl-CoA can also be a product of HMG-CoA. So if we wanted to produce a little bit of energy, like if we only want to produce 20 ATP of energy, we'll just take acetoacetate here and throw it into the blood plasma into our peripheral cells, and we'll have it go up this little chain, which I'll get into in a second. But if we wanted a little bit more energy, like a reference right here with 22.5, we can reduce acetoacetate into BHB right here. So we are tossing on a hydrogen over here and reducing acetoacetate into BHB. So we'll just start from the ground up. When BHB flies across the blood plasma into our peripheral cells, it is just going to basically work back up the chain. We are going to go from BHB and we are going to become oxidized. BHB is going to become oxidized and drop off that hydrogen to NADH right there. And with this NADH, guess what? We can go through a little redox reaction right here. So this is where that extra 2.5 ATP come from, because we can go through a little redox reaction here, because all of these cells have mitochondria, and where the mitochondria are found, that is where redox reactions can occur. So 2.5 ATP can be produced by this little step right here, and BHB can be transformed back into acetoacetate. And then acetoacetate will go and be acted upon by succinyl-CoA, and succinyl-CoA will drop off a CoA onto this acetoacetate and turn it into acetoacetyl-CoA, and we'll get our succinyl as a little product right there. And then acetoacetyl-CoA will have another drop off of a CoAsh right here, and we'll eventually transform back into two acetyl-CoAs. Those two acetyl-CoAs will go through the TCA cycle and produce 20 ATP. So if we get reduced all the way to BHB and come up through this entire process, in all reality, we are producing 22.5 ATP. But if we stop here at acetoacetate and fly through the membrane right here, we are only going to produce 20 ATP because we do not get this low redox reaction right here. And we do need to know a little bit more about this. When ketone bodies leave the hepatocytes, or flying over here, they are acidic and will react with bicarbonate ions in the blood. And if we remember bicarbonate ions from last try, bicarbonate ions are basic components. So if these basic components are reacting with ketone bodies, our blood pH is going to lower, because our amount of bicarbonate ions is going to decrease, and that will put us in a primary alkali deficit, which is basically saying our blood is more acidic than it should be, and we will eventually progress into metabolic acidosis if this condition is going on for a long time and eventually becomes chronic. So metabolic acidosis is just again telling us that we are in a chronic state of more acidic blood. And when it refers to ketone bodies actually causing this, it will be referred to as ketoacidosis. Yeah, and here's some steps that go a little bit more in detail about a lot of these processes. I'll just leave that reading for you guys, because again, I have been talking to you guys for a while. So I'm gonna end this video. And again, I apologize for being low energy today, not as enthusiastic. I still love this stuff, but some days are better than others. So keep on killing it. I wish you guys the best of luck on your quiz, and any questions, again, shoot me a text, give me a call. I am happy for everything. So have a great day, everyone.